Hi, this is Dr. Simon Says, and this is a response to npage85, which I have been debating with for quite some time, and I've realized it would be pointless in continuing this debate without explaining some pretty basic stuff when it comes to DNA and basic chemistry, which npage85 is completely unaware of. I don't think he has passed high school chemistry, to be honest. The things he says are quite ludicrous. So npage85, look at this video, try to learn something, and then we can continue discussing this topic. I'm going to link in the description box some earlier videos where we are discussing DNA replication, and in one of my videos I say that DNA is capable of replicating itself and that we have the evidence for it, and npage85 questions this evidence. This is the article I linked to. It's called Chemical Self-Replication of Palindromic Duplex DNA, which can be found in Nature. And this is the response from npage85. I have read it and it seems to be fairly obvious, yeah right, that it isn't DNA they got to self-replicate without proteins. Facepalm. So there's still the problem of DNA replication being irreducible complex. Okay, and page 85. Not the response I had expected. I thought you had some basic understanding of DNA. Let's get some definitions here. Well, we can look at what the article says. It says, here we report the self-replication of palindromic symmetrical duplex DNA like oligonucleotides, 24 monomers long, in the absence of enzymes by means of a cycle that transfers information from template to copy and is potentially capable of extension to include non-symmetrical sequences, selection, and mutations. Well, I think the word you got hung up here is DNA like. For some reason you started to think that this wasn't DNA, it was some other form of mythical, magical, synthetic molecule that had nothing to do with DNA, or that, well, I'm not really sure what you're getting at, but let me explain a couple of things. When we're talking about nucleic acids, we're usually talking about RNA and DNA. Those are two big groups of nucleic acids. Nucleotides are the basic building blocks of nucleic acids. They are made of a nitrogen-containing purine or pyrimidine base linked to a sugar and a phosphate group. An oligonucleotide is a linear sequence of up to 20 nucleotides joined by phosphodiester bonds. When we get greater, they will be called polynucleotides instead. And beyond that is DNA. So let's look at the oligonucleotide. Here you see we have the base, we have the sugar, and we have the phosphate backbone. Now let's look at DNA. What do we have here? We have a base, a sugar, and a phosphate backbone. So up here we have DNA, and here we have oligonucleotide. So what is the difference? Well, the DNA is double-stranded. So what they did in the article I referenced was that they made an oligonucleotide, which is a base, a sugar, and a phosphate. And then they made a duplex DNA copy. That is, they turned it into a double-stranded oligonucleotide, which is exactly the same thing as a DNA molecule. Only it's very short, and therefore it was easier for them to make it replicate in the absence of DNA copying proteins. Now let's take a look at the basic chemical reaction. Here we have some double bond chemical reactions with H2. As you can see, both reactions are similar, or the mechanism is almost identical. However, the starting molecules and the end products are not. But it makes no difference. We're only looking at the addition of H2 to a double bond, which will reduce the double bond to a single bond and add the hydrogens to the carbons. Now, the only difference here is the time frame between these two reactions. So, a catalyst is a substance that accelerates a chemical reaction, but is not consumed or changed in the process. Now, you argue that a catalyst sometimes is needed for a chemical reaction to occur at all, which is somewhat true, but not true at the same time. Let's look at another picture. Here we have an energy diagram of reactants to products. In order to go from the reactants to the products, you must overcome the free energy. And here we see the uncatalyzed reaction. It's a much higher energy required to produce the products, whereas you have a catalyzed reaction, a much smaller amount of energy is needed. So the time frame is faster. This reaction will happen much faster, much quicker than the uncatalyzed reaction. Now, sometimes this activation energy is so high that the time frame for it to happen is close to infinity. Therefore, we say that the reaction is impossible. But that's not really true. Then we use a catalyst to reduce the energy needed for the chemical reaction. Do you understand the difference? I don't think you do, and I'm looking forward to hearing your response.